We're going to continue on with our last part of this unit on protein synthesis by talking about how does a protein actually get made. So my hope is that in this unit I've expressed how important proteins are to a living organism. Everything physical, for the most part, now some things are carbohydrates, some things are fats, but for the most part, most of the physical components of an organism are going to be proteins. And cells, for the most part, are protein factories. They produce all different kinds of proteins, depending on the type of cell. Your liver cell is going to make different proteins than your skin cells are going to make. And your stomach cells are going to make different proteins than your bone cells are going to make. And that makes sense, hopefully, because they have different jobs. But the thing I hope you remember is that they're all using the same master copy of DNA. If you think about it like the cookbook analogy that I keep using, every cook has the same cookbook with all the recipes that ever existed, but each cook is going to use different recipes based on what kind of restaurant they work in. So that's how your cells work. Your liver cells and your stomach cells are using the same DNA. It's just that they're using different parts of the DNA to make the proteins that they need for that type of cell. So what we're going to talk about today is, all right, well, how do we actually make these proteins? And it is a two-step process. And today we're going to talk about the first step. And the first step is called transcription. So what does transcription mean? Well, the word to transcribe, right? Transcription means to transcribe. What does transcribe mean? It just means to copy. If you transcribe your notes, let's say, for example, I'm talking right now. If I say, all right, I want you to transcribe what I'm saying, you're just copying it down. You're not changing it. You're not putting it into a new language. You're just copying it word for word exactly what I'm saying. And that's what's happening with transcription in your DNA. The information that's in your DNA has to get copied by your RNA, specifically your mRNA, so it can be taken out of the nucleus to be made into a protein. And that's all that's happening during transcription. The mRNA is copying the DNA. So let's start talking about it. So what is transcription? Very simply, it's the process in which mRNA is made from DNA. And I'm going to explain the steps now, but it's almost exactly like replication. So if you remember back to DNA replication, helicase came along, unzipped the DNA strand, and then DNA polymerase attached to the strand, and it added the appropriate bases. So at the very end of the process, you get 2 new strands of DNA. The entire strand of DNA got copied. That is not the case with this process. In transcription, only the gene that is being made gets copied. So I'm going to go back to my cook analogy again. If I'm a cook and I want to copy a recipe out of a cookbook, do I copy the entire cookbook or do I just copy the recipe I want to make? Well, if I am doing things the right way and I care about my time, I'm just going to copy the one recipe. And that's what transcription is. This specific protein needs to get made, so the RNA copies just the code for that one protein. And if you remember, in our DNA, we have 30,000 genes. So there's 30,000 recipes in there. And if the DNA was copied every time, the entire thing, that means 30,000 recipes have to get copied instead of just one. So there's some big differences between DNA replication and transcription, but when it breaks it down, they're very similar to each other. So let's talk about how they're similar. First of all, where does this happen? This happens inside the nucleus. So this process happens inside the nucleus, and specifically, it happens in the nucleolus. So the nucleolus is where the DNA is being stored, so this process happens there. So how does it actually occur? Well, the first thing that happens is we have an enzyme, and that enzyme is called RNA polymerase. Now, that sounds very familiar. In replication, it was DNA polymerase. This is RNA polymerase, and that's because it's copying RNA. It's doing RNA, not DNA. But what it does is it attaches to the DNA strand at a place called the primer. Okay, so what is a primer? A primer is a 3-letter code that tells the RNA polymerase where to attach. How do I know if I'm reading a paragraph where a sentence starts? Well, the way I can tell is there's a capital letter, right? A sentence always starts with a capital letter. That lets us know that it's a new sentence. Well, think of the promoter as a capital letter in a sentence. It lets you know where the sentence starts. Now, I know the period tells you, but let's pretend there's no periods in this scenario, and it's just capital letter. And that lets you know where it starts. So there's a specific codon is called the promoter, and that tells the RNA polymerase exactly where to attach to. So that's the first step of this process. The second step of this process is the RNA polymerase is going to unwind the strand and separate it into, it's going to basically open the book, right? It breaks those hydrogen bonds and it opens up an area on that DNA strand that is going to be copied by the mRNA. One half is going to be used as a template. The other half just doesn't get used. So one half gets copied. It's going to be converted to mRNA. The third step is the RNA nucleotides are going to match up with each base. And don't worry, I'm going to explain this and go over it a little better than this, but I'm just going through the steps first. So the RNA nucleotides, so for example, if it's A on the DNA, the RNA polymerase is going to add a U, because remember, there's no T's in RNA. If there's a C in the DNA, the RNA polymerase is going to add a G, and that's going to go for the whole length of that gene. How does it know when to stop? Well, the way it knows when to stop is, how do you know when a sentence is over? Well, there's a period or an exclamation point or a question mark. Well, this has something called a stop codon. And a stop codon is a 3-letter code that lets the RNA polymerase know when it's time to stop copying the DNA. Now, if that stop codon wasn't there, it would just copy the whole thing. And that would be a big problem because it's not supposed to copy the whole thing. So just to go back to what I use as my analogy here, if I have a sentence, I know when the sentence starts because it has a capital letter. And I know when it ends because it has a period or an exclamation point or a question mark. So think of the promoter as the capital letter and think of the stop codon as the period at the end of the sentence that tells it when to stop. So I want to show you an animation real quick that will hopefully help you to understand this.
Egg proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. Motor region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand...
So this process is actually pretty simple. It's a lot like replication, but the big differences are it only copies the section that needs to be copied. It doesn't copy the whole DNA strand. It just copies the section that is coding for a specific gene. So they're going to be different lengths depending on the protein, right? Longer proteins are going to have longer mRNA. Shorter proteins are going to have shorter mRNA. How does it know where to attach? Well, there's that thing called the primer or the promoter, and that's that little code that tells it where to attach. And then there's the stop codon that tells it when to detach. Now, what happens to that mRNA once it gets copied? Well, that's the next step, but that's called translation, and it's going to leave the nucleus and it's going to go to the ribosome. But we're going to talk about that in the next lecture. So it's just a quick review of what I talked about today. We have a process in which a section of the DNA is going to be copied by the mRNA in the nucleus, specifically in the nucleolus. It's going to copy that length of that protein, and it's going to know where to attach because of that primer or promoter region, and it's going to know where to detach because of the stop codon. We have our special enzyme called RNA polymerase, which does the job. It unwinds the DNA, and it copies whatever it needs to copy by adding the appropriate RNA base to the strand. Thank you very much.